So good evening everybody, and welcome to the Coleoptera Society. Today we're not having the business meeting. The business meeting is going to be online on the 13th of December. I hope to see lots of you online on the 13th. But today we're only going to be having the talk, which is going to be a beautiful talk by Bob Anderson, entitled The Weevils. He's going to tell us about what might be the most diverse group of Coleoptera, or might not be. After this, we've got a few minutes to finish the sandwiches, of which there are many. And then we are going to adjourn to the Sheraton, where Anthony, the treasurer, has organized a small tab behind the bar. If you want to order drinks on the Sheraton Coleoptera Society tab, you have to ask the bar staff for Anthony's tab. Now, we haven't said Coleoptera Society or anything general like that because we don't want everybody mooching in and ordering drinks on the society. So it's called Anthony's tab. So I'd like to hand the floor over, please, to Bob Anderson from the Canadian Museum of Nature, who everybody here knows as a leading authority on the Cuculinoidea. Thank you very much.
Thank you, Max. I'm going to turn on the record here. I've been a member of this society for 44 years now. I joined in 1980, and it's just like a second family, the professional family aside from your biological family. So it's always great to stand in front of this crowd and talk about beetles. I gave one of these talks in 1984. Milt Campbell and I, that's 40 years ago. Milt Campbell and I, up in Toronto, we talked about the Queen Charlotte Islands. Milt was talking about staphylinids and I was talking about weevils. So this is my second talk, and it took 40 years for them to invite me. I want to talk about the weevils tonight. The title is sufficiently general so that I have the latitude to talk about anything I wanted. I wasn't tied into talking about anything about biology or phylogeny or higher classification or anything at all. A couple of things before I start. I've pilfered slides from lots of places. Steve Marshall has been very generous in letting me use a lot of his photographs, but I've also taken various ones from the internet, okay? So none of the photographs are mine. I am not a good photographer, so I'm relied on other people to illustrate the talk. With that, I'm going to start off. I want to dedicate this to Charlie O'Brien. Everybody here knows Charlie. He died a number of years ago, 5 years ago. And Charlie was very, very generous to me. When I was a young student in 1981, I think it was, I spent about 3 or 4 weeks at his house. And Charlie told me, he said, anything where there are 10 specimens and I have a species name on it and there's duplicate data, you are welcome to take a pair. And that would establish a reference collection that you could use so you would get to know the weevils in the very, very broad sense. Lots of people had specialized in particular groups, but I think Charlie saw in me, like himself, this general broad interest in all weevils. And I came back from that trip with about 40 mailers filled with weevils, and that was the basis for me understanding weevils and getting to know the weevils as a whole. If I had not had that opportunity, I would not be in the position I'm in now, and would not have the knowledge that I have now about weevils as a whole. So I'm really, really, in favor of what Charlie did for me at that time. It was a wonderful gesture. Even though he did serve some time in the Chilean mafia during his time in Chile. So I want to talk a little bit today about how I got started working in weevils. It's a little bit maybe of an oversharing story, but what the heck, I got the podium, so I'm going to tell you the story. I want to talk a little about classification, diversity, and then I'm going to get into, instead of talking about any one particular thing, I'm going to bounce around and talk about some really cool things that weevils do. Most weevils are associated with plants, plant products, Some weevils, though, venture a little further away from that and do some strange things. So we'll talk about those and the morphology that's related to that. Talk a little bit about current research that I've been working on, and I want to just say a few things about what I see as the future, where we're going with things. I did my bachelor's degree at the University of Toronto. Before that, I had been a bug nerd. When I was a little kid, my mom would always pull out my pockets and out would fall all these kinds of insects since I was about 5 or 6 years old. So I always knew I wanted to do something with entomology. So when I went to the University of Toronto, I took a systematics course with Glenn Wiggins, who was a trichoptera specialist. And Glenn overheard me one day, I guess, saying I wanted to go on to grad school. He pulled me aside and he said there are 4 groups that he would recommend that people work on. And because they're sufficiently diverse that you'll spend a lifetime working on them and you won't ever be short of questions to ask, there will be opportunities for you to explore relationships with other organisms, be they host or parasite or wherever it may be. So it adds at different levels of complexity, which allows you to tell all sorts of neat biological stories. And the other thing is, they're all somewhat economically or extensively economically important. So if you don't get a job doing pure science, you can get a job doing something that has more of an applied angle to it. So he's looking ahead from those perspectives. So he had suggested mites, parasitic Hymenoptera, chrysomelids, and curculionids as the groups. 2 of those we could knock off right away. Chrysomelids just didn't appeal to me. I've heard many people say, and many in this room say, you have to really love and relate somehow to the organisms that you study. And chrysomelids, sorry Sean, sorry Ed Riley, sorry other people who work on chrysomelids, but weevils were the ones that cut it for me. So weevils were the group I picked. I wanted to do a master's degree with Henry Howden up in Ottawa. Henry was the professor at Carleton University, and his wife was an expert on curculionids. And while Henry would be my official supervisor on paper, Anne would be the de facto supervisor that I'd sit down with every day and learn all about weevils. So I went to visit Henry, and Henry took me into his office and said, I just accepted 2 other grad students, a fellow named Bruce Gill, some of you may know, and a guy named Al Ritchie, who was going to work on cynipid wasps. And he said, but, he said, across the hall, Stuart Peck has just been accepted on the faculty at Carleton. Go talk to him. So I went and talked to Stuart. Stuart, however, had money to work on sylphid beetles, carrion beetles, but he didn't have funds to work on weevils. So we sat down and we worked on a project where I would do my master's with Stuart on sylphid beetles, and then my plan was to go and work with George Ball at Alberta on weevils. So after completing my master's, I went to see George, and George had, if you knew George, it wasn't all about your marks. It wasn't about, anything academic. You sat down with George over a glass of scotch, and he talked to you, and if he liked your passion and your general knowledge and your devotion to the subject matter, he would take you as a student. So that's how I got in with George. We drank a lot of scotch that night. I convinced him that we were a great group. Did my PhD with George, got an NSERC, which is the Canadian government postdoc, and decided I was going to take it somewhere in the States. And I was going to either go with Charlie in Florida at the time, or Horace Burke in Texas. And it all boiled down to this book on the lower right-hand side. When I was younger, I got this book in the mail one time, and it's a story, it's all these little cities in Texas and all the scenic things to do in Texas. And I would leaf through that book, and it was called Texas Lived the Legend. So I had to go and live in Texas. So we went to Texas. So I was dating this gal named Catherine at the time, and she said, what am I going to do? She says, if you move to Texas. We'd been dating for about 5 or 6 months. And I said, you could come with me. And she says, well, yeah, but only if we're married. And I said, yes, exactly. And she said, is that a proposal? So off we went to Texas. So that's how I got interested in weevils. Probably oversharing, but anyways, it's a little fun story. Classification of weevils. As Max mentioned, they're probably one of the, if not the most diverse group of beetles that we know of, although the staphylidid people are giving us a run for the money these days. The weevils are broken up into 8 families. There's the Cymbridids, recently proposed as a family level in a molecular systematics paper by Shin et al., where they recognize the Cymbouridids is the sister group to all the rest of the curculionoids. The Mnemonichids, the Anthribids, Samantha working on those, the Bellids, Atalabids, Carids, the Brentids, and the Curculionidae. The bulk of the diversity in this group is in the curculionids. I'll get to that in a few minutes. At the top here, you can see the, up here, you can see the Cymbouridids at the very top as the sister group to the rest of the curculionids. Then you can see the Mnemonichids, and down into the Anthribids, the Bellids, the Atalabids, the Carids, the Brentids, which is a big hodgepodge group, and the Weevils. Within the Weevils, there tend to be actually three main groups. There's a group of primitive Weevils that would comprise the Brachycerines, the Dryopthorines, and the Platypodines here through the central area in yellow. And then we have the curculionids that most people are familiar with. The broad-nosed Weevils, the curculionines, the molotines, all these other various groups, including the scolatines as well. They're divided into two main clades, the CEGH clade and the CCCMS clade, named after the subfamilies that they encompass. So Cymbouridids. Cymbouridids are the pine snout weevils. They are associated with pines, with conifers. The adults are found on the flowers, and the larvae develop in the strobili on the flowers. So they have this very, very tight association with pine trees, with pines, a conifer. Mnemonichids have a similar association. The Cymbouridids, there's only about 15 or 20 species, mostly north temperate things, in both the Palearctic and the Nearctic region. Mnemonichids have one group that extends up through the Americas to high elevations in the southern US, but then they're mostly southern hemisphere things, and they get on to Araucarias and conifers related to Araucarias. The one that we get up into North America, Atopomacer, the males have this big projection between the front cocci, and they're associated with high elevation pine trees. This is Pinus culminicola on Cerro Potosí, where we collected a bunch. And yes, there was snow on the ground when we collected them too. These things tend to be active really, really early in the year before the snow melts on the ground, as soon as the pine flowers come out. Anthribids, fungus weevils. Some of these things are quite large, look like cerambicids with long antennae in the males, some down to these mite-mimicking species that are only a millimeter or so long. These things you usually get in the leaf litter, and a lot of these other things are found on fungus on dead logs as well. Bellids are an interesting group. You have sort of a conglomerate of species that have different biologies. This one here, Rhopalotria, is associated with cycads. The males have these big and large front femora. Oxycraspidus is associated with Araucarias in South America. Rhinotia is associated with acacias, interestingly, in Australia. Balanophorobius I described from Balanophorus, which is a parasitic plant that has no chlorophyll, from Costa Rica. And this one here is a thing Adriana Morvaldi and I described from specimens we collected on the llama trip, the leaf litter project. This one was from Nicaragua, and it's called Archicorinus. We don't know what it does. We collected it just beating out of some dead twigs. But this is a conglomerate of things that is associated with these plant parasites, with cycads, and then with twigs of acacia, and then probably dying stems here. But a very, very interesting group. Atalabids, divided into two groups. We have the leaf-rolling weevils here on the left-hand side, and the flower head clipper weevils on the right side. These guys are so called because they'll lay an egg in a flower head, usually of an Asteraceae, and then they'll clip around the base of the head of the flower, kill the flower droops over, and the larva develops in the dying flower. These guys here are the ones that roll leaves into a little tube, lay their egg inside, and then the larvae develop inside. This is a thing called Trachelismus, which is from Southeast Asia, from the Philippines. And goodness knows what happens with such a thing. This is a male. The females have a very short neck, and perhaps this is somehow involved in some type of sexual selection or combat. But it's a really, really cool beast. Caridae is an interesting group, a family that's relatively young, named for the genus Car from Australia and some of the Southeastern Asian areas, Papua New Guinea. They're found on various conifers. in that area. Very small group, not very well known at all. There's one group that gets into Chile as well, also in conifers in that group. The bellids are an interesting group, and this is a hard group always to characterize. You have your typical bellids here, elongate, narrow underbark things. This one probably has an association with ants in dead wood. This is cycas, or Silas, the sweet potato weevil. Here's a species of apion. If you look, the one character that unites all of the brentids is that if you look, the first two abdominal ventrites are on a different plane from the last three. So it's almost like you walk along the first two, and then you step down to the next three. And all brentids, Silas, apionines, and things have that same character. So that's the one thing that pulls such a amorphous group together. A couple of others here. Antleorhinus is another brentid. The male here with the short snout, the female with the long snout. The female, we think, uses this long snout to dig into the cycad seeds, and then she turns around and she lays her egg into the hole that she dug with her rostrum. The New York weevil here is a bit of an enigma. It's variously Ichthyceras novoboricensis. It's been given family status as the Ichthyceridae, but here we group it now with the brentids. We're pretty sure that's where it goes. And so apionines here across the bottom. This is the hollyhock weevil, male on top of the long-snouted female. A different species here, a knee apion species here, and a nanophyid over here. You'll notice that in all these primitive weevils, the antennae are straight. They're not geniculate. They're not bent, elbowed like in true weevils. But nanophyes are an exception in that they have geniculate antennae. But they're obviously apionines because of the structure of the trochanter of the legs. The trochanter is like a cylinder that is a separate, complete segment between the femur and the coxa. In other groups, the tip of the femur actually touches the coxa, and that way you can tell them apart. So once you see that character, they're obviously apionines. Now if we go to the Curculionidae, as I said, the Curculionids break up into three main groups. A primitive group consisting of the Brachycerids, Dryopthorids, and Platypodids, and then these two clades, the CCMS clade and the CEGH clade, and then the Bagawines, which are kind of a little bit left out. We're not quite sure where they fit. There are a couple of different classifications you may encounter for the weevils. One will, the Curculionidae, one will include these families, or subfamilies, the Brachyceridae, Dryopthoridae, and Platypodinae. And that one is one where you can define the Curculionids by the presence of geniculate antennae. If you choose to move the family down and exclude these as separate families, then you can use the structure of the male genitalia to define the family. But here we use the characteristics of the antennae. But you will see both options in various classifications you might see. So that's just the simple difference between the two. So Brachyceras, these are Brachycerids. These are large weevils that occur in the circum-Mediterranean area. They get up to about 3 or 4 centimeters long, some of the big ones, associated with monocots. Arorhine weevils are another tribe within the Brachycerinae. These are aquatic weevils. This little Tannyspirus lemnae on lemna pads here, and this one here which gets into Equisetum, Gripus equisetae. It also includes the Raymondionimids, which are the blind weevils. These things have primitive genitalia like the other groups. However, they're very small, very depigmented, and they are blind, completely eyeless. Nearly all members of this group are eyeless. The Dryopthorids, they tend to have antennae that are situated right near the base of the rostrum, and the scate goes back over and passes by the front margin of the pronotum. They also have a very, very distinct antennal club, which has a very little hairy apical part, but then a very, very shiny, hard, chitinous basal part. And that's usually the best ways to recognize those. This is 2 species from Vietnam. These guys get to be pretty big. Protocerus colossus here can come up to about, well, I guess about 80 or 90 millimeters long, so about 8 or 9 centimeters. This is probably the biggest weevil here. You can see a couple illustrated with the wings out. Some of them have quite long front legs, as you'll see there. Platypodines here as another group, strains the pinhole borers. These are interesting and enigmatic, and people have traditionally classified these with the scolotines, but in fact they are closely related to the Dryopthorids. Molecular systematics brings them out usually as very, very close to the Dryoptorids. Okay, the CCMS clade, I want to whip through this a little bit. This is where, you know, between the CCMS and the CGH clades, it's sort of the bulk of the weevil diversity is split between the two. This is where you get all the little weevils that are associated with plants. They tend to have a very, very small apical hook at the tip of the tibia, which they can hang on to the plants with. That's in contrast to the other group where you start to get very similar looking weevils, but they have a much, much larger hook because they tend to be associated with more woody plants. And Thonomine weevils, such as this one here, one of the plum curculios, although that could also apply to a species of Conotracheus. You can see the long snout, you can clearly see the geniculate antenna in this guy. These little leaf miners here, this Tachygonus lecontii, which mines the leaves of oak in eastern North America. It's a cool little thing. Big hind legs, they do not jump. They look like little bird turtles sitting on the leaves. Cossamine weevils, under bark, tend to be black, shiny. Very, very few of them have any kind of scales. They'll have hairs and things, but they're generally this morphology, and they vary in size from about 5 or 6 millimeters long down to about 1 millimeter in length, and they're horrible, I find, to work with. Pseudorhynchine weevils. These are neat little weevils. Some of them jump, and some of them break away from the traditional life cycle of weevils, where in most weevils they're endophytic. So the adult will oviposit inside some part of a plant, and the larvae develop inside that particular part of the plant. But some pseudorhynchines feed externally in the leaves. They create this gummy substance, like a slime, and they move around in that on the surface of the leaves. So they're a little bit different that way. Baridine weevils, this is a couple of the more colorful baridine weevils. A lot of these are, the baridines are very black and monotonous, difficult to separate. Associated with all sorts of different kinds of plants. Some of the baridines have these neat little spines in the males. In some cases it'll be a single spine, and some it's a pair of spines. And there's been some studies done in Costa Rica where they joust back and forth with these spines, the males. They only occur in the males. So again, we see some kind of sexual selection going on. So there's all these neat little, peripheral studies you could do with respect to weevil biology and the things they do. Here are some molotine weevils. These are the ones that have a bigger spur at the tip of the tibia that they used to hook onto the various plants with. This is Pisotes here, the pine weevils, and a species of Rhizomatus marginatus from Argentina over here on the right. I think they're associated with acacias fabaceae. Cryptorhynchines are a huge group in the neotropics. We call them the hidden snout weevils because they tuck the snout into a groove between the front legs, and there's a little cupule right here into which the tip of the rostrum fits. And that little cupule is developed from the mesosternum. Okay? There are other ones that have a channel that go along here, but there is no cupule, and those are in different subfamilies or different groups. Lixines, these tend to be big weevils, usually in dry, arid areas, although the genus Lixus occurs a lot in marshy areas. Hard to characterize except for the large size, but one of the things they all possess is if you dissect a female, there are these paired symbiont sacs along the side of the reproductive system, and they house bacteria, so that when these things lay their eggs, these symbiont sacs coat the outside of the eggs with the bacteria that then the developing larvae uses to help it digest the food that it's eating. We actually think there's been some studies to show that this is quite widespread in the weevils. We just don't understand it fully, and we don't know the mechanisms by which it's transferred. But these guys have these really neat bag-like structures to do that. And the scolatines. A lot of papers you will see deal with weevils will say the Curculionoidea excluding the scolatine. That's not because we don't like them or anything, it's because traditionally they've always been sort of kept as a separate group. So a lot of people who study weevils in the very strict sense know very little about this, so we leave that part of things up to Sarah and Anthony and the other bark beetle people. Okay, the CEGH clade is the other large clade here. Entomine weevils are the large group here. These are the ones that are most of the introduced species. A lot of them are parthenogenetic, so there's females only. They travel around very well, they establish easily. They have a short snout here. The adults feed on foliage and they lay eggs that develop in soil. Usually the larvae feeding on roots, but externally they live in the soil. Or in some of them they create like a little pocket along the side of the root in which the larva feeds. But they have short snouts. If you look close up, here is a photograph of the tip of a rostrum of a recently emerged entomine, and you can see they have these processes by which they break out of the pupal case, and they use these to dig through the soil to emerge, come to the soil surface. And in the process of doing that, they usually break off, and when the beetle is mature and has emerged, you get these scars, which are the leftover spots where these broke off. So that's one way you can always tell an entomine, is that there will be those scars. And these processes have various different shapes and forms, and some can be quite large and small and hooked and the whole bit. There's quite a bit of morphology associated with those. Cyclamines are a difficult group to characterize. There's some aquatic ones. There are ones that are in southern South America. They're a big group in Australia, in the desert areas of Australia. Bagus is that problematical group. They're aquatic weevils. They have a sheen, like a waxy coating almost across them, and they're aquatic. They do go down into the water and swim around, and they have like acts like a physical gill that helps them stay underwater. They're very much like some of the primitive water weevils that occur in the aerorines, but they have advanced male genitalia, so they fit somewhere else into the system, so we're not quite sure where those go. This is from a paper that a few of us, myself, Rolf Oberpreeler, and Adriana Marvaldi did in 2007, sort of looking at the diversity of weevils. And you can see here it's got the numbers of species across the top. It's a slightly different classification from what I presented earlier, but you can see that things such as the Adelabids have about 3,000 species, I think, but the bulk of the diversity is in the weevils. And there seem to be a number of sort of key adaptations that have taken place. There's the evolution of a rostrum here. There's the evolution of using the rostrum for oviposition here, where the female will dig a hole with the ovipositor, turn around and then lay an egg in that hole. And that gets her egg away from potential predators, parasites, desiccation. It puts it right inside the plant where it has to be to develop. Then you get the elongation of the antennal scape, so that the female, when she's burrowing the hole, can pull the antenna back in and line it up alongside the rostrum, and it gets out of the way. And then you have also related to that, you have the evolution of geniculate antennae here, characterizing the curculionids in the strict sense. So there appears to be a series of key adaptations that have taken place throughout the history of the weevils in association with the evolution of angiosperms as well. I mentioned about using the snout as the ovipositor. This is an example of a curculio species, acorn weevils, and here you can see a female on the left. She's got her rostrum buried in the nut, where she's going to then turn around as she does on the lower right, and if you look down in the right here, you can see the ovipositor extended, and she's actually laying an egg into that hole that she's created with the rostrum. You see incredible sexual dimorphism in a lot of the weevils. This is a species of Conotriculus, I named after Terry Irwin. And this is a female. You can see the amazing length of the rostrum that these guys have. They must be in some kind of big fruit. This is another species I just talked about, this Philorostris. You can see again, this is on a big Annonaceae fruit, and the females use this rostrum to bore their holes into the fruit. And I bet if you dissected these, you'll see that the ovipositor is like a big long telescoping tube that could fit into that hole and push the egg right down into the very bottom. Mimicry, I want to kind of diverge from talking about some of these specific things and just talk about some neat things about weevils. I want to talk a little bit about coloration and mimicry. Here in some examples from the Philippines, on the left here is a species of Cerambycid in the genus Doliops. Then you have a species of Pachyrhynchus and a species of Metapocertus, a different genus, all converging on that same body form. The Philippines is just incredible for diversity. I once asked Charlie O'Brien what his favorite weevils were, and he said these guys here. He called them the clown or the Smurf weevils. Here's 3 species of Pachyrhynchus, 3 different ones from different places, but just this incredible mimicry that is taking place, this convergence in scale patterns between these things. They're also mimicked by Alcidotes, which is a molotine. Here you can see some of the metallic Alcidotes that have these patterns that converge. It's just an amazing system, and we don't see anything like this in any other parts of the world. It just seems to be strictly in the Philippines. The colors are created by these very, very small scales that are in kind of just slight depressions on the surface, and it creates these amazing patterns, just absolutely beautiful. You can see why these things are really popular with the hobbyist collectors. These are extremely popular with that group. Eupholaus is another genus from Papua New Guinea and Irian Jaya. Beautiful coloration associated with sweet potatoes, apparently. And just spectacular colors, and probably about 40 or 50 species known from Papua New Guinea. We've started now, myself and an Italian fellow, Maurizio Bolino, have started looking at the internal sac of these. He takes the genitalia, male genitalia, and he inverts the internal sac, the endophallus, and he expands it so we can see all the different lobes, and it's just opened up a whole other set of characters for these things that allow us to explore the relationships and the systematics of these in a more informed way that doesn't just rely on color patterns. I mentioned in that EGH clade, one of the H stands for the hyperines. I didn't show any pictures there because I had some pictures here. This is one of our North American species. It's actually a European thing. It's introduced into North America, but Hyperapostica, the alfalfa weevil. Very simple looking brown thing. But when you get to Madagascar, this is what you get in the genus Chloropholus. Like, look at those spectacular colors. Are they mimicking lady coccinellans perhaps or something? I don't know, I just find that incredible. Here's a photograph of a living one on the left. And on the right here, you can see this is a larva of a hyperine here, and they're external on the leaves. They feed externally on the leaves. And here's the cocoon up here. It's like this loose-woven silken cocoon that sits on the surface of the leaves. And there's a genus and species that occurs in Central America, and it's called the golden cage weevil, because it builds these bright golden cocoons of this loose woven silk that surrounds the pupal case, which is inside. This is a specimen I just saw at the Field Museum a couple of weeks ago. Had to take a photograph. I think this is what weevils must feel like after they wake up tomorrow morning, after a night of drinking. So, genus Desmophorus. These are some of those brachycerines. They're very, very primitive things, but they have a much longer stem than these guys do. Some fantastic ones occur in Madagascar. Lithinus is another strange weevil that occurs in Madagascar. Madagascar has some strange stuff, I'll tell you. These things mimic lichens, and you can see here's an individual here, weevil, and over on the right you can see the same weevil sitting on a twig which has the lichens on it. Just incredible camouflage and crypsis for these things. The Insectarium in Montreal has a little display where there are five or six of these on a little twig, and they dare you to find them all. They also tend to have, this Lithinus also tend to have little plants, mosses, lichens, and things that grow on the back. They tend to be long-lived, and they live in very, very wet habitats, and they get these little gardens that grow on their back. There's a genus called Gymnopholus, which occurs in Papua New Guinea, and there's 2 lineages. The lineage on the left has a roughened elytra, and the one on the right has a very smooth elytra, and the one on the left has these little gardens of things that grow on the back. And in fact, some of the specimens have actually been found with little rotifers that live in the little pits, the little water that collects on the backs of these things. And Lindsley Gressett was a famous entomologist who did a lot of work in the South Pacific, and he coined the term epizoic symbiosis for these weevils. But they're really, really neat things. This is a thing that Chris Lyle, myself, and Joanna Cristobal at the British Museum have been working to try to describe. But in Colombia and Ecuador in South America, you get these weevils that are, typical black dead wood feeding things, but they have these rows of little pustules that look like little mites on their back. And we don't know why this is the case, but this is one example here. Here is another example here that you can see it moving along, but look at those big pustules on the back. This is something undescribed. We're trying to get some specimens out of Colombia so we can get that thing described. That's just one cool weevil in my mind. Waxy wonders. A lot of weevils, when you collect them, particularly in the genus Lixus, which I showed you earlier, have this pollinosity that accumulates on the surface. And we think it's a wax exudate. It might act to conceal the thing somehow. Maybe it's got some other role with pheromones or something. We don't know exactly what its role is. But in some weevils, you get extreme cases where the wax grows, as this. Maybe these things, it's disruptive, coloration in some way. Maybe they don't look like a weevil because they have such an irregular outline when you see them from above. But these guys here from Southeast Asia have this waxy protrusions that stick out. And in the extreme version, we get this fellow here, which is described from New Caledonia by Willie Kuschel in the early 2000s. And here is the body of the weevil, and here's this incredible wax growth in this big horn that comes out. I've never seen one like this. We have a couple that I've seen which have this part broken off right here, but I mean, it just isn't that spectacular. I just think it's wonderful. Okay, dung weevils. I don't know how many of you know that, but there are weevils that feed on dung. And of course, where else do you think it would occur but in Australia? And this is a genus called Tentigia, a fellow named Hermes Escalona, who is now at CSIRO, and he's their weevil expert, the new weevil expert. He just revised this group. Here we have a kangaroo. You see kangaroo, rather than cows, kangaroos produce nice little round pellets that are all ready for the beetles to roll. And Tentigia weevils have taken advantage of that. So here's a short video that Hermes let me use, which shows you a Tentigia rolling its little dung ball. And this apparently is the first time this has been recorded, but you'll see the weevil here in a second rolling its little dung ball across the ground. So you can see that it grabs a hold of the dung and pulls it. They take them apparently underneath a rock or underneath a log, and they stash them there. Then they lay the eggs on them, and the larvae develop inside the little ball of dung, which is of course mostly plant material. So that's pretty cool. Australia is not known for its dung beetles, but it's sure famous for the dung beetles. Weevils are also associated with mammal nests. There's a species in North America, you get it commonly down in Florida, Hormops abducens, and they're in tree squirrel nests. They're not associated in any way with the dung or anything that the squirrel does. They're there simply for the concentration of all those twigs. Apparently the larvae develop in the twigs. Down here we have Cabiaphylla, which lives in the burrows of the guinea pigs in South America. And this strange thing here, Epiphytophytes, which we're not quite sure what it does. It turns up at lights, but it's closely related to these guys and probably also is somehow associated with mammals as well. Ant associates. Weevils are associated with ants as well. Jack Longino, who's here in the audience, and Jack collects ants, and we've known each other for quite a while. He was in Monteverde and collected some weevils out of the stems of an ant plant called Triplaris. And there's a species of Azteca ant that lives inside the stems. And he got some weevils living in those stems that had been abandoned by the weevils. And so I spoke to a fellow I know down there, many of you I'm sure know him, Angel Solis, who was a former curator at Indio for the beetle collection. And Angel and a couple of friends went out, climbed the Triplaris, cut thousands of stems, cut them all open, and we got about 50 or 60 weevils out of those. So that was really cool. Got some larvae as well. This is the weevil, probably represents a new genus of Conoderine that Henry Hespenheim, myself, and Jack are going to work on describing. Here in Arizona, I've been after the people who run the ant course every year down at the Southwest Research Station to try to find this thing, Lyometopholis manni. It's a little cryptorhynchine weevil, and they live with Lyometopum ants. But they're rarely collected. I've only seen one series of these that Carl Steffen collected at Cochise Stronghold down in southern Arizona. This is a neat story here. In some of the cosynines, these are cosynine weevils, there's a genus called Acanthus, this one here. And Acanthus you can get by sifting the dead wood at the base of an old magnolia tree, and you often get lots of Camponotus ants in with the weevils. I don't think the weevils are living with the ants, I just think they're living in the same habitat. But then if you move down here, this thing, Prionarthrus, it's only known from one species from Brazil, but we have them multiple species now all the way up into southern Mexico. Large serrated outside of the tibia, the tarsus recesses into the tip of the tibia, they walk on the tips of the tibia. You get this one here, which shows all these modifications for sort of trying, perhaps living with ants. But then you get this thing up here, which has all these same modifications, but then it starts to have the trichomes, these glands that appease the ants. This is undescribed, these are all undescribed, so if anybody's looking for what I think would be a fantastic thesis project, would be to look at the evolution of these in association with the ants that they live with. Mike Ivey and other people have fogged these members of this group out of trees in the West Indies, and we think they perhaps live with some of the ants that build carton nests in the trees. Long appendages, lots of weevils have long appendages. These cryptorhynchines from Fiji have these incredibly long antennae. It's probably mate guarding. They probably stick these big antennae out and they guard a female and they turn around and they make sure no male comes within mating distance of that female. So it's a way of they ensure their paternity. I know that's what longhorn beetles do. I'm pretty sure that's what the anthribids with the big long antennae do. But that's what I think these guys do too, although it's never been seen or documented.
Oh my
You
God.
get these fellas here, this Talanthia. The species name is Phalangida, of course, you would expect. And the males, again, have these big long legs, and perhaps they stand over a female, and they guard the female, which is laying her eggs. Again, this Trachelismus, which I showed you earlier, this Adelaide, which perhaps has this big long neck because of sexual selection. Here's one of the Talanthia, photographed in Vietnam or in China, southern China, and you can see that the name phalangid is very appropriate for these things. You also get these guys, which is a Dryopthorid Mahakemia campmenteri from Java, and here's one here. These are big weevils, and you can see the long front legs of the male, and I would bet, while the biology has never really been studied in these, I bet these guys sit over top of the female, which has very short front legs, and patrol a radius around her to keep the other males away. Blind weevils. A lot of weevils are blind and they get into caves, and you're thinking, what the heck are they eating in caves? And I think they're feeding on roots that come through the ceilings of the caves. That's what I think they're doing. They tend to be in groups, such as the entomines and other groups, where they're feeding on roots or dead wood. This is a species, or a genus Baldorhynchus, from the Central European area. Lots and lots of species in Europe, but up until a few years ago, we had none in North America. So I had contacted a fellow named James Riddell, who was a cave biologist. He worked on carabid beetles. And James sent me all of their weevils. And in that, we started to find some species that were associated with caves. So I had revised this genus called Lymantes in the southern states. And you have a species here in the middle, which you can see a very big eye. There are some that have just a few eye facets, and then there's one that has no eye facets. Here you can see on the left, you can see a thin row of eye facets in this one, which you get in leaf litter all through the hill country in central Texas. But this one here is only known from caves in central Texas, and you can see it's totally blind. I had thought there was only one species, but later on we found specimens of a second species. This one is north of the Colorado River in the Edwards Plateau area, and this one is south of the Colorado River. But two separate species of blind Lymantes that live in the caves. So we do have cave weevils in North America, but they're not near as diverse as they are in Europe. Another project I've been working on was this genus called Dicozenus in the southwest. It's a broad-nosed weevil, short snout, feed on roots. And this one in the west and on the left is a typical one you get in desert areas on the vegetation, probably at night. Over on the right is a species that's only known from one individual from the mouth of a cave. But this guy, Mark Sanders, who's in charge of the endangered species programs down in the the central Texas area in the Edwards Plateau, he went into this cave in the city of Austin called Spider Cave, and he got this thing. And he got multiple specimens. Apparently there's one big rock in Spider Cave, and if you look around the sides of the rock, you find this thing, but it's got reduced eyes, long, thin appendages, and depigmented, clearly, that you can see here, you can see the deciduous, these cusps on the tips of the mandibles. But I think this is just a species of that genus Dicosinus that has become highly adapted to caves. If I were to recognize this as something different, then I got to split up Dicosinus all over the place. But I just truly, so there are these really cool cave weevils in North America. I'm just amazed, and I think if we keep looking, we'll find more of these things. For many years, probably those of you who know me best, is that my goal has been to push all of Central and South America through a Winkler funnel and get all the weevils out of it. That's been my objective. So over the years, I've collected in many different places, and I really love collecting these leaf litter weevils. I just now hope I have some time to do something with them. I revised this genus Steagnetti in 2010. There was one described species with 2 syntypes. We now have almost 100 species in a paper I did in 2010, but we keep getting more every time we visit a new site. And on the Llama project, we probably have added about 20 or 30 more. But I mean, we're not splitting hairs here. We're not having to look at molecular sequences or anything to tell the differences. Just look at the incredible variation in external morphology in these things. It's, the weevil diversity truly is incredible. Limantines, the group to which Steagnetti belongs, are all characterized by having the eye out on the snout. And here's 4 different genera from Mexico and Central America. This is the only one here where the genus is described. These other things are all new. Multiple species in these as well. It's not just like this is a single species in that group. We might have 5 or 6 species that share that basic morphology. Here's a plate Vasily Grebennikov and I put in a recent paper sort of documenting limantine diversity, although we didn't name anything. We just showed this to show the amazing diversity of these things. It truly is incredible. I want to talk a little bit about iNaturalist. At the ECN meetings, Ingolf Askavold gave a nice talk, and it was a nice symposium, a short symposium arranged, talking about the role of iNaturalist. This is a species of Toxorhinus, a dryopthorine weevil from Ecuador. And I saw this picture on iNaturalist, and there's only 2 other species in the genus, so I thought that thing's new. So I ended up contacting the author, the photographer, Thomas Shahan, and he had more pictures he could let me use, but he did not collect specimens. But I got in touch with some people in Ecuador, got some specimens to describe the species. But the first place I saw it was on iNaturalist. And I think more and more people are finding that now. You have so many eyes out there looking for things. We've documented new records for Canada, new records for the United States, new provincial records in Canada, all through specimens we see first on iNaturalist. And we look at it and we say, that's not something I recognize, and then you explore it further. Sometimes the people who see it will actually go and collect specimens for you. It really is great. Now, in 2017, I'm going to close up with a couple of quick things here. 2017, I wrote a blog for the museum about the extinction of the greater chestnut weevil, Curculio caryatropes, in North America. As we know, the American chestnut tree is functionally extinct. There's hybrids that exist now, but none of the original trees, as far as I know, were producing any nuts. And I would have bet, and the focus of this article was that this weevil was extinct. It was gone. And it's the biggest Curculio we have in North America, beautiful big thing. Well, this time last year at the ECN meeting, Michael Charles, who's been working on this as a master's program with Dwayne McKenna in Memphis, he came up to me and he said, I found, this is Curculio caryatropes here, sorry, this is the female on the left, the male on the right. Beautiful Curculio species. He came up to me and he said, this picture here was just taken in the Longwood Botanical Garden outside Philadelphia. And what is it? It's a Curculio caryatropes. And this one here now we know is from Delaware. So these were posted on iNaturalist, and through those, we've now tracked down that this thing lives in 3 different places, and it's on American Chinese chestnut hybrids. Okay, it seems to survive on those. But again, this role of iNaturalist that we see that's really playing a big role here in things now, and I think will play a bigger role as we move forward. I just want to finish up with just some thoughts about the future. In Weevils, I think we need to continue work to resolve these in-family relationships. I showed you the cladogram with the CGH and the CCM, the 2 various clades. We're still trying to work out relationships. We need more material to resolve those further. I feel that where I'm going to continue to work and where I can make the biggest impact in the rest of my career is trying to describe species diversity, trying to work on descriptive taxonomy. Let's describe and name some of this diversity, because there's so much of it out there, and I think we really need to get it named before we may lose that opportunity. I think we're going to see museums as increasingly important sources of material. I've heard many people talking here how difficult it is to get permits now, how difficult it is to ship material between Europe and North America, restrictions that are put on us now to move specimens across borders and so on. And I think we're going to see going into museums as increasingly important sources of material. One of the things I want to do when I retire is to go to various museums and look at their specimens and try and spend a couple of weeks at each museum and sort all their unsorted weevils so that it's available for the next generation to study. I think we need to continue to develop in-country expertise. So we need to encourage people in various countries throughout the world that they need to develop experts in particular groups of insects, weevils in particular, in my case, to increase their knowledge and their ability to identify and study those things within the country. I've seen these graphs which show the diversity of life throughout the world, and it's skewed. All the diversity is in tropical countries, and all the people who study it are in the temperate countries, and we need to make that graph more equitable. So we need to encourage this sort of study. We need to recruit students, okay? I know we have a number of great students that are working on weevils, and we want to see more continue to work on weevils. And I also see a big role for social media and citizen science moving forward. Yes, there are problems with identifications. Yes, there are problems with those photographs from 40 feet away of the insect sitting on black wool. Anything about it. But we can work around those things and make the kinds of finds that we saw here. So with that, I'm going to end, and I'm going to say thank you very much. It's always a pleasure to address this group. Thank you.